Information Management and Applications from NTHU in Taiwan, an MSc in Cells and Systems Biology from U of T, and then a PhD in Bioinformatics from UBC. She now works as the head of data science in the bioinformatics section at the National Microbiology Laboratory, where she applies data science to microbiology, genomic and proteomic data sets and tackling biological problems. Please come in. She's the winner of the 2019 prestigious Canada Trophy for the International Data Analytical Challenge. And when a pet project does not consume her spare time, she's outdoors enjoying nature. Julie will present her adventure shifting from mammalian to microbial domain and working with different types of biological and omics data. So with that, we'll join me in welcoming Julie.
Thank you very much for the kind introduction, Terry. If you would like to find a seat, feel free to grab one in the front. So today I'm here to share with you a little bit of introduction on microbial omics. And I'd really like to thank Sandra and Terry, and as well as Vicky, for setting up the seminar. This would be a little bit of my personal experience. As you can tell, I've already exhausted my educational options. So I wish I could study more because there's always more to learn. But yeah, so I'm now applying my skill set at NML. So all of the stuff that I present here is from myself. I'm not representing the government at all. I just work over there as a public servant. So I'd like to congratulate in celebration of the 50th year anniversary for Faculty of Science at U of M. Looking at the departments, you can see biology, comp sci, stats, math, all these different departments. Although they're distinct, these are really interdisciplinary. There are a lot of connections between different departments. On top of that, we recently have the Nexus Data Science, which really connects data sets from different domains. So personally, my background comes in from math, a little bit of information systems, bioinformatics, and molecular biology. Aside from the education, generally I've interacted with a lot of different experts from different domains, from stats, math, comp sci, biology, and now microbiology. Personally, I do not assume any expert domain knowledge. I come from the bioinformatics background, where I apply data science to omics data sets, essentially. It is very important to work with domain experts. For those who are budding scientists, you'll see a lot of examples of how these collaborations gets done, and sometimes it's like playing volleyball. We toss the ball to another person with the finding, and the biologist may try to validate it if they believe it. So convincing the biologist to do it, it's another story. Right, so the title is Adventure. Personally, in my life, I feel like adventures are things that are new and interesting, and these are experiences that you may share with others and also involve learning. Whether you're traveling foreign country, it's learning a new culture, etc. But importantly, we have to live to tell the tale, because if you look at the definition of adventure, it involves danger. So this is a photo of myself and two of my friends snowshoeing at minus 26 degrees Celsius in 2017. It was a photo from Winnipeg Free Press. They even asked for our consent. It was my friend's first time snowshoeing, so it was fun for her. We were learning, and we saw deers running by, and I was still pretty new to Manitoba. I didn't see deers that often, so that was really a mini adventure in Winnipeg. The same thing goes with omic data science, where we are constantly addressing new challenges. That is, if we solve the previous one, or we'll be addressing the same challenge with different aspects, perhaps using different types of data sets, and we're always learning and applying, and we collaborate, right? We learn a lot from our collaborators because they are domain experts that in the domains that we don't have expertise of. And ideally, you would have some sort of discovery and validation. And sometimes science doesn't always give you positive results. But I would argue that even with negative results, we learned things from it as well through the process. Now, I just have to talk about common struggles after talking about adventure. Oftentimes, communicating to different people in different domains, it's like talking in your second language or vice versa, right? Like, just like my presentation, there may be certain things that go over your head depending on your domain. I was told to try to make it as broad as possible, which I will try. But there are always jargons that we may not be aware of, domain knowledge, and oftentimes the same term can mean different meanings in different domains, and then the same different terms can have the same meaning. This communication bridge just makes it difficult to really get into a different domain, and we can always get confused by who to seek help from. If you're not familiar, maybe you would think, oh, I need someone from the stats department. But what type of stats? Oh, goodness, I have no idea. So that's why we have interdisciplinary training, where we have new trainees and with different, perhaps 2 or 3 domains that they're working in. And that way we learn the languages of each other through grad school or through taking courses. And that really helps quite a bit. The only thing I'd like to draw to trainees' attention is that there is a trade-off between being jack of all trades versus collaboration. Essentially, in grad school, we try to learn as much as we can all about our project, incorporating different things, but there is value in collaboration as well, and when you get into the real world, that's where the projects really shine. So that balance in terms of proportion will depend on the nature of your position and project. So I will finally talk a little more about science. Firstly, it will be on omics in biological science, then getting into where data science may fit in, and then moving on to omics data science in microbiology. Because prior to joining NML, I was mainly working on molecular biology in the mammalian field. And then lastly, I'll conclude with career information and some tips for the trainees. So I don't know if you've ever seen this movie. When I was a child, I thought it was really inspiring. This is called Gattaca, which is from the DNA sequence. It's a movie about a society that uses genetic screening to determine the upper bound of your career. So it turns out this Ethan Hawke, in fact, has genetic heart defect, but was able to achieve and become an astronaut by taking somebody else's identity. So that just goes to show it's a fictionary thing, but it's really inspiring in terms of we can potentially have higher potential above our genetics. And now we know there are epigenetics as well, and all the environmental influences. So if we get to the modern knowledge of genetic variants, there are genetic disorders which can be really serious, where Nick, the motivational speaker, he has Win3 gene mutation, and that led him to be born without 4 limbs. There's also genetic predisposition, the susceptibility, where you've seen news from Angelina where she has BRCA gene mutation, and that results in higher risk of developing cancer sometime in her life. And so that's the type of things we're dealing with, and oftentimes with the information from genetics that helps us understand the relevance to health. Just to keep a little bit of perspective, there are currently less than 8 billion people on Earth. Does anyone know how many cells are in our bodies? Any wild guesses? No? Trillions of human cells. And of course, the majority of each of the cell would have the set of DNA, your chromosomes, and then it's passed down from your parents, so you'll have the variants on the insides, and that sort of becomes our genetic fingerprint. But let's not forget there are a lot of microbiomes as well. The microbiology with microbes, fungi, bacteria, viruses, those are actually trillions of microbe cells as well. There's a debate about the proportion, but I think scientists would agree on the association of these, both human cells and bacterial cells, to our human health. So I don't think I really need to go into the definition of genes, genetics, and genomics, but genomics essentially is the technology and the study of the entire genome. Because we now have a much better technology to actually screen our genetic material at once. In the past, there may be genetics tests with PCR in small scale. There is now genetic genotyping arrays from Illumina, which gives you greater than 600,000 SNPs. On top of that, we now have whole genome sequencing. So that gives us a lot of information. This would be information for one person, right? And if you think of the entire genome and multiple individuals, how do we know which SNP is associated with what trait, what biological trait or disease? Generally, if you look at genetic counselors or those commercial tools where they would essentially look into ClinVar, SNPpedia to get information. And these are databases that came from scientific experiments. There are a lot of brilliant minds working on it to generate publications which gets curated into databases. And so that involves biological studies, wet lab experiments, maybe on specific model systems. Statistically and analytically, one could take the set of data sets and test for associations in order to find what are the variants associated with the trait. But of course, in biology, we want to have that validated ideally, because that's where it gets curated into database. Now that we know how the data sets comes through, one of the analytical approach would be to essentially, like I said, test for association. And in biology, oftentimes we're comparing between groups. So in simple terms, let's say a bunch of cancer patients, a bunch of healthy individuals, we want to see what are the genetic predisposition for a particular cancer. Then you could look into the genetic material of all these individuals and find what are the variants that are associated with. Now, once you have that, you can arrive to a lot of SNPs. Another thing one could do is to look into the biological mechanism. So this leads more into the molecular biology aspect of things, where we want to sort of learn to interpret those variants. Especially in mammalian genomes, you have 97% of non-coding genome, and that's really hard to interpret. And usually these SNPs would not be in the coding genome, because otherwise it may have detrimental effect. The person may not be existing to have that data sets obtained. So a little bit on the stats part of things, where the genome-wide association study, in this particular example from the Wikipedia, you could have patients, 4,000 patients, maybe cancer patients, and 6,000 non-patients, the healthy control. And here what you would be getting is looking at first SNP, for example. What is the frequency of G at first SNP within the group and within the second group? And from this case, you can actually test using chi-square test to find the significance of association of the SNP to your trait, right? And then you can do so. This is one simple example, one simple approach. You can do so for all the SNPs. Remember, we had 600,000 SNPs that you can get from an array, right? I mean, cut down with some filtering, it may come down to a little less. But nevertheless, we have to remember to test, to adjust for multiple hypothesis testing, because that's a lot of tests you're doing. And I see Sandra nodding her head really nicely. So we will get variants associated with the disease trait afterwards in terms of the significance level. So here's the one example that you can draw from literature. You see these type of plots a lot, where the x-axis represents the location of this particular SNP, represented as a dot in the figure is on the chromosome. And then the y-axis is the significance level. It's minus log 10 of p. So the higher the y value, the smaller the p value. And because there is an adjustment of multiple hypothesis testing, you can see that this line for significance, sorry, it's a little hard to aim, that's the adjusted p-value threshold to declare significance in this case. And as you can see, there are so many different dots that are actually significant. So what would a biologist do? Like, do you test them all to actually really understand the underlying mechanism? That's a lot of headache, right? So that's why I mentioned that one can do further analytics, pulling in other information to try to narrow down and interpret these variants. especially they are generally in non-coding regions. So in this case, I've actually done a project on it where I tried to interpret the GWAS variants, and these involved utilizing transcription factor predicting binding sites, as well as chromatin interaction data sets. So I will get to a little more of that later on, but this is one example where you can utilize other information and bioinformatics to analytically try to make sense of things. So in this paper, I essentially proposed an analytical approach to interpret these variants. In this case, this particular variant from the A to G, there is a difference, an increase in MYC, which is a transcription factor binding site. There's a higher affinity over there. And in fact, this particular SNP is near the ROR1 cancer-related genes. This project, unfortunately, wasn't validated, but I'll get to a project that has biological validation. So that was part of the genomic annotation, like making sense of things in the non-coding genome. And the next project that I'll talk about would be on predicting enhancer regions. So in our genome, there is a lot more things that goes on than just the genetic sequence. There are promoters, enhancers that can be really far away, still regulating the gene. There are transcription factors, epigenetics. So with all these different biological mechanisms, there are different technologies to use to interrogate the cell and generate data sets from it. So there's genomics, epigenomics, transcriptomics, and proteomics. Right, there's also other omics, which I haven't dabbled in, so I won't get into that. But what are these, why do they exist? Because the genome gives us potential, whereas the transcriptome, proteome, give us the more higher closely to the function and the phenotype of the cells. And with all these massive data sets, that's where data science can come in place to really try to make sense of the data. So just to get to the enhancer identification project, that's where in 2012 we published this project where I started in 2011 and 10. The simple term is that I essentially find the bins in the genome and allot the potential enhancers and promoters that we think are highly probable and use them as training. And I had a third category, which is unknown. In retrospect, I should have named it something else. But essentially using the genomic features, CpG islands, and epigenetic data, et cetera, cofactor binding sites, that comes into a structured data set. If you're more in the machine learning or data analytical realm, you would understand that. So the columns would be features, the rows are the genomic bins. And since, like I highlighted in that toy figure, there are a lot of regions in the genome that we don't know the function of. And those ones would be gray, and that's not used for training. It's just left out because we want to build a model, then predict those regions in order to understand it. So now here, this is called supervised learning. Essentially, I want to learn the pattern, what are the important features that separate these different classes, and then when there comes a new sample, which is a new bin in gray, I want to be able to predict it. So may I just have a show of hands of how many people are familiar with machine learning? Oh, that's quite a lot, awesome. So I'll go very quickly. Essentially, you've got an instance like the matrix that I've just shown. The key thing is we want to leave out some samples, right? Because when you build a model, you don't want to overfit. And one way would be five-fold cross-validation. There's another thing called out-of-bag. But what it does is essentially you're using four portions to build the model and then predict on the left-out portion, which we call the validation. Ideally, you want to report it with the test set, but generally speaking, you can depend on the approaches to obtain the feature importance, and then you can assess the model performance, right? And if we're confident with it, then we'll use it to predict new bins. So I'm not sure how much I want to get into this. So oftentimes, statistically, you can try to be a minimalist. We do not use too many features, because in the case of omics, in fact, there are a lot of data sets, a lot of features, like let's say the variants, you have a lot of them. And if you just purely do the stats, then sometimes if you incorporate them all, then you can overfit, right? Whereas this lasso regularization essentially narrowed it down, and it infuses this penalty where a feature has got to contribute to the model and reduce that model error. Otherwise, we are not going to give it a weight. We're not going to give it a microphone to speak, essentially. So then we'll just ignore that feature, right? So that's part of it, which gives you a minimal model that hopefully performs well. And in this case, and oftentimes I would say with analytics, we want to double check with biology whether that makes sense. And in this case, I can see CpG island contribute really positive to promoter, which makes sense. And P300, which is a cofactor, contribute to predicting enhancer, and that makes biological sense. So there's always sanity check. And so I'm just going to fast forward to the particular example that I circled up there. There is SOX2 gene, and there is a set of different enhancers that I predicted to be highly probable that's greater than 100 kb downstream. And so these enhancers, in fact, have been validated. The students after me actually validated using enhancer assays to make sure that, in fact, they are essential for SOX2 expression, so even though they're really far apart. And these are the things that we can overlook, but utilizing bioinformatics and tossing the ball back to the biologist, if they believe it, they can be validated. Right, so back to school and work. So that was me beforehand, before coming to NML. A lot of these work are on mammalian cells with genomics, transcriptomics, epigenomics, et cetera. Now, working at NML, I'm more in the microbiology realm and utilizing genomics, metagenomics, and proteomic data sets. That would be the examples that I talk about. And this, again, is a little more for the trainees who may be curious. So at work, I do quite a bit of consulting and also R&D. Consulting is also providing the service because I'm at the core facility. Usually, I would consult analytically to colleagues or train their trainees on data analytics and as well as data visualization and interpretation. Myself could also be doing design and implement, for example, proteomics data analytics and use that to help support the researchers. On top of it, at times, there would be trainees that do not do scripting. Then I also created a tutorial utilizing graphical user interface tools so that they can also do the analysis themselves and maybe consult with me just to make sure that interpretation is right. And another thing is, just like any job, we want to anticipate the needs too. So we can't just do whatever work that's going on. There may be ideas that I may try to follow just to do a little bit of R&D myself, and we want to provide resources. On top of that, I'm curious as a person in general, so I also do general club presentations every month or to every other month. That way we get literature update to see what goes on out there. And these are the things that, the type of things you would do as a grad student anyways when you're doing the projects. So I'm just gonna go ahead and show you some figures that I generally generate and look into either for omics, transcriptomics, or proteomics data analyses. So it always comes down to, oftentimes you're comparing two groups. There can be more elaborate and complicated design, but this is an example of comparing groups. Let's say you have 3 on 3, and then a lot of data sets require normalization. You want to make sure the bias gets adjusted, and then you would do dimension reduction, which applies to a lot of other data sets than omics. And we would look at the differential abundance analysis of genes and proteins. On top of it, with bioinformatics, once you have the differentially expressed genes, you would want to see what their functions are. Then you can tie it back to the biology. So it's a full circle. Then you can chat with your collaborator and see if that makes sense. And that's how the whole project can get pushed further, right? On top of that, if there are enough data sets, then I would do supervised machine learning at times, just out of curiosity, even if we are not interested in model prediction, but it's always a fun exercise just to see how far we can push it, right? As I mentioned, there's the human genome-wide association studies. There are also bacterial version of it. And the context generally could be source attribution. Let's say there's a salmonella outbreak, and you sequence the genome, and you want to see, wait, did it come from the chicken or come from, cows? Like what type of foods that ended up giving you the contaminations? So that way, you could also look into, if you have a base of the sequences that you already know, then you can do supervised machine learning. First, to identify what are the variants associated with more present in chickens, which are more present in cows, et cetera. And that type of existing building knowledge will allow you to predict new samples. That way we can sort of get a sense of potential to see where that isolate come from. By the way, isolates are terms for bacteria. So in that setting, you can actually also do machine learning as well, because usually with GWAS, you need a lot of samples. So another application would be to look into antimicrobial resistance, where you compare whether the bunch of isolates are more resistant to this particular drug, or are they more susceptible. So as you can see, just with similar structures, although every data can be different, there are always catches in biological data sets, but just looking at the structure, you can do a lot of things if you actually have a system of analyses. So just like with ClinVar database, NIPEDIA, for AMR, there is the Comprehensive Antibiotic Resistance Database, the CARD from MacArthur Lab in McMaster University. This is where they curate a lot of AMR determinants. So these are genes, also some SNPs that may be associated with certain drug resistance. So in fact, one could essentially utilize that database to screen your genomic information and to sort of predict whether this particular isolate with that genomic information may have resistance to certain drugs, right? So this comes to a small project that I did. This is utilizing public data sets that my colleague generated, Dr. Cliff Clark. I essentially utilize it to look at it from the antimicrobial resistance angle, and we found some interesting things, so we decided to publish it. So that's just adding an analytical capacity screening for AMR. So generally with antimicrobial resistance, if you're not as familiar, they are phenotypic testing where you would measure the MIC, but there's a lot of multi-drug resistance, then you have to test either a panel of different drugs, or if your interested drug is not in the panel, you have to do different tests. And it involves culturing the cells too. Whereas genomics, you can essentially grab the genomic sequences, run through whole genome sequencing, and utilize that CARD database and screen through it, sort of like how we screen for susceptibility for Angelina Jolie's case. Now, what I came in was to use the proteomics to essentially do a one-stop screening using the CARD database as well, because proteomics are closer to the phenotype. And so there's no doubt in my mind that there would be cases where maybe there are some genes that are present, but it's not expressed, right? There's also differential expression. Like I've worked with transcriptomics data all the time. So because when I utilized my colleague's data set in Campylobacter jejuni, it was 4 isolates, and he had genomics and proteomics data sets, and I screened with CARD with RGI and proteomics, and I found discrepancies. That's why I started nudging him, asking, hey, have you done AMR on your isolates? And so he ended up contacting the AMR group and then generated these data sets. So I just want to draw to your attention that indeed there was one case where, even in the small number of subsets, there was one case where the genomic presence is not the same as protein abundance. So that goes to show if one were to utilize genomics to do this AMR screening, we may predict it differently from proteomics. So here's the case for OXA61 gene, which confers resistance to beta-lactamase and ampicillin. What you will see here is that in genomics, there are 3 isolates with gene presence. It's in red, and 99.6 is the percent identity to the CART database. Whereas in proteomics, you only see the protein abundance in the last isolate. And when you look at the phenotypic testing, only the last isolate have the the values raised higher. And furthermore, if you're interested in molecular biology, if you chase back to the promoter region of the sequence, when I look into that, there is in fact a mutation here, a variant at this position in the last isolate that differ from the other 2, and this creates a Tata box. The way I knew it is because I looked it up. There was a paper that in fact did all the biological characterization of this T allele at that location. Oops, did I? Right, so that was the story of this one, where analytically, if you're in the analytical science, with close collaboration with your biologist, the research scientist, you can still try to push the boundary a little bit. And then it's like, again, tossing the volleyball, where if it's interesting enough, we can chase after it to really look into it. So that adds to the capacity within the proteomics core facility, where we can actually do that search regularly nowadays. And another example that I'll mention is that oftentimes it pays to pay extra attention to the data set. This is an investigation of chlamydia infection using proteomics. Like I said, I would build the pipeline analytical process, and oftentimes you would think, okay, Julie, you've built it. Let's just all run through all the data sets through it. But every data set can be different, and it can be because of the experimental design or the biology behind it. So this is one example that I want to show you where the biology behind it is driving a bias which needs to be adjusted. So chlamydia is an intracellular obligate bacteria. And essentially, when you extract proteomics, it's different from other bacteria. Like when looking at Campylobacter, you're only looking at Campy's proteins. But with chlamydia, because it has to grow in the host cell, when you extract proteins, you're extracting both the chlamydia as well as their host proteins. So in this case, my collaborator are looking into comparing with plasmid, without plasmid of chlamydia infection versus no infection. But because the P+, with chlamydia, the infection, in fact, has really active chlamydia, and the ratio of the host to chlamydia is much lower because the chlamydia is just thriving, whereas P- isn't such a way. And so in this case, when we look at the protein levels, I'm just going to go very brief on this, which is on the y-axis. If you look at the distribution of the yellow part, it's much higher than the P-. And that's because you have a different proportion to begin with, right? So if you look at it, it'll be almost like, oh my gosh, all proteins are definitely significantly upregulated, if you're familiar with the volcano plot, because there's just that scale. And when we do the normalization, we used everything, right? That's why I added specific species-specific normalization. That way you bring it to scale, because we know for a fact that initially there are different proportions. And in that sense, then that brings the volcano plot down to much normal looking. And here you can see, in fact, there's a protein in chlamydia that's actually upregulated when there's no plasmid. And that has been validated with Western blot. So that's essentially something that gives you comfort that, okay, that adjustment is appropriate. And of course, I went through to look in adjustment to the human side and then test for a functional enrichment, which I won't get too much into. But essentially, generally, when we do analysis, we still want to think of the biology, right? Even if I don't understand, then I would be interacting with the experts to make sure that these are making sense. Okay, so next up, I will switch gear into microbiome, which is essentially a population of bacteria, fungi, or virus in one. And this is looking at gut microbiome, where machine learning is applied to try to classify the label of the samples, which is whether there's certain immune diseases or whether these are healthy samples. I'll keep it really brief because I want to skip to the next chapter. So with fecal microbiome, my colleague generated these microbiome data sets that measures the microbes within the fecal matter. And you've got a set of individuals from healthy control with their microbiome and a set of individuals from Crohn's disease, etc. And the question is whether or not we can actually tease out the microbiome patterns between different cohorts, right? Different diseases or versus healthy. And when there comes a new sample, are we able to predict the correct label? So again, if you listen throughout the talk, I'm trying to make it a theme where it's always the comparison and we're trying to separate it, and the features that you're sort of sorting through are different. There's a lot of front-end processing involved that can affect your results, but I won't get too much into it. So essentially, I built up this microbiome analytical workflow where you've got the data sets, and I, in fact, got it from my colleague, and where you have these OTU counts, which are the taxonomy. You can think of the bacterial taxonomy, what type of species, actually to the level of genus. And of course, there's always filtering and normalization involved. And in this case, because they had technical replicates, biological replicates, and we could look into different things where how far are the distance, how similar are the technical replicates, how similar are the biological replicates. And it's good to check these things for sanity check. And that's another dimension reduction approach, MDS, where we tried to look at all the samples and you color-coded, lay the color about the labels on top of it afterwards. Now, I passed that through random forest model, which is building a lot of trees to inform decisions, and then look into the feature of importance within the model. And that is something with biology one can look into, okay, what are the important genus that actually separates from healthy versus Crohn's disease, for example. And then we look at the model performance, comparing different diseases, because I tried multi-class machine learning model, but it didn't work as well. Essentially, it's because, for example, here, the MS versus multiple sclerosis versus rheumatoid arthritis, the pattern were quite similar, and we were not able to separate it as well. So I just will lastly touch upon the CAMDA challenge that I did. This is relevant to the microbiome data, but this is in urban setting where samples from different cities are used to predict to collect it to obtain the microbiome data set. And this was at the international conference, and it was really interesting that it was a lot of people sharing. But I want to give you a heads up, this is actually a negative result, and yet we still won the CAMDA trophy because a lot of people worked on this problem, and it is a difficult problem. The reason it is difficult is that the challenge from last year is, can you predict the source of this microbiome sample from your trained cities? Like if you've already collected samples from the cities before and bring that into your model, it's a lot easier to predict it be correctly. But if you want to predict something that you haven't sampled before, that's very difficult. And I'll just show you how difficult that is. So the question marks are the mystery samples that they pass to us so they can evaluate our performance. Of course, there's a lot of front-end processing involved my colleague, Dr. Andrea Tyler, joined on the journey, and she did the front-end processing, and the front-end processing makes a huge difference. I don't have time to show that here. In fact, using different approaches gives you different sensitivity of the results, and that affects your model for sure. So if you lose information, then your model would not be as good. Now, what I did was to use multivariate regression approach where I predict the longitude and latitude because it's a new location, so I wanted to free myself up from training using classification because we are always going to predict wrong location because these are new ones. And in fact, I hate to say this, but it didn't improve much. And of course, when you do cross-validation, you're mimicking prediction of origin for samples from trained region already. Because cross-validation, you randomly leave out some samples, chances are you already have samples from the same cities in the training. Whereas if you use a cross-validation, leave one city out, then this is the performance. On top of it, I will just quickly look at the classification. It looks at the diagonal, meaning that the reference to prediction, they're mostly accurate. Whereas if you leave out a city and don't use any samples from that city, then train the rest of the model, in fact, the performance for predicting the new city isn't as great. Like you can see that the challenge over here. But in fact, you can sort of see that they come from the same continent, but that's still, again, a little stretching it. I think I'll just stop here. And so in general, I hope you get a flavor of the omic data science from the human side and also from microbial side, where every data set, in fact, can be a little different. Sometimes we do need to customize our workflow and pipeline. And with data science, really, we can use the data set, same data set, sometimes with different perspectives, right? Yes, biology can generate different data sets with different perspectives, but some of these involves collaboration, and I hope that I stress the importance of domain expertise. So with that, I'd like to acknowledge all the majority of the researchers that I listed up here for working with me. I really learned a lot from all the collaborations and things. I mean, obviously not enough because I wouldn't know all the different details of the domain expertise. That's why we collaborate, right? But again, it's sort of like really like playing volleyball. You're trying to pass it and not let the ball drop. And it really takes attention for multiple experts. Lastly, I just wanted to show you that we have at work, there is NML NML science stories. And the microbiome study was one of it. But if you're interested in what goes on in NML, you can search for the keyword and then go into the link, and you can see a lot of different studies that is explained in lay terms to the public. And so here I'm getting into the tips for trainees. One thing is we want to know ourselves, our limitations, our strengths, weaknesses, and we stay curious and active and try to provide value to the group and to the society and humanity as a whole. Of course, in the process, we want to maintain the work-life balance, and then ultimately, we want to give back where we take. So before I go, was there any questions, or can I just go through continually? There are just a few more slides for, yeah. Okay, so here are a little bit of my experience of networking and opportunity. I'm just going to flat out and say when I was in grad school, it's a lot of work. I didn't have time to go through all these other functions. But that's because now I'm working, so I presented at Meetup, the Winnipeg Data Science Meetup. If you're in data science, it's really great. I'd highly recommend that. And we have Carlson presented there too. And so there is LinkedIn. I've heard people finding positions on LinkedIn, Twitter. I know people actually post on there, and you can get really latest scientific updates. There's also Toastmasters for people like me. I've been working on it through giving better presentation and also learning about leadership. And as for grad students, to find these connections, oftentimes it's conferences, workshops, and collaboration from your advisor with others, then that way you can reach out to different universities, and also internships if you can find any. As for undergrads, I actually see our past co-op students here today. Thanks for coming. So U of M has a really great co-op program, right? So it's great to take use of it, and that way you get a snippet of sense of what it's like to work in certain environment. And plus, if you're capable, you can show how well you can perform, and maybe they'll be wanting you for the job too, for later positions. And there's also FSWEP, which is federal involved too. And then if you're interested in a career in the government, there's jobs.gc.ca. So there is an email subscription if you sign up, and you can, the key thing is when you're applying, make sure you examine the posting in detail. What are the requirements, how you can stretch to it. And I just have to warn you, these are long applications. I took mine took 10 hours. I mean, I'm not ashamed to say it, I got the job. It took me 10 hours to write the application, and because I wasn't applying for jobs beforehand. And also, it's a long process. If you see inventories, for example, those are things that are preset. They may use that, draw people down the road. So I would highly recommend if you see these, and if you're interested, you find a position that you like, definitely try to apply as soon as possible, because you can actually update your information for inventories. And so this is small fonts. This is one example. If you search for keyword for data, there's data science positions. And if you search, you can also search keyword for biologists, and these are positions. Right? So if you subscribe with email, then you don't have to monitor it all the time. There are also other resources for undergrads, such as post-secondary recruitment, and there's a Twitter, Canadian government jobs online, and of course there are also provincial government opportunities. Lastly, I hope I can inspire some data science students. There are oftentimes different data science challenges. And so there, of course, you would have some challenges from companies where they really want your scripts, right? So that's a little bit selling your script to them, where if there's a prize involved. There are dream challenges, these are more biomedical related, and the CAMDA challenge, they're holding that this year again. So it wasn't just microbial genomics, there's also drug-related ones, as well as another one is cancer-related challenge. Lastly, there's aqua hacking. In case you haven't heard, there are things that in the Canadian public that really requires data scientists' help, and also people with technology engineering experience to try to solve problems, such as phosphorus in our lake. So that's it. That's it for my presentation. Thank you for your attention.
Julie. So I'm sure she would be more than happy to answer questions either on acquiring government jobs or through the omics or anything that was presented today. So with that, please feel free to ask questions. And then after that, next door, we have our usual meet and greet with food and beverages in the SSA lounge. Any questions for Julie?
Yes.
So in machine learning, for a specific task, we usually have a lot of different models which can do like the job like easily as good as the other one. Like what sort of parameters do you use, especially for your research, in being like the best model to use?
Right, so it really depends on the data sets, the type of question that you ask. It's a golden question, right? Like we want a panacea, like we want something that's perfect. I've heard people doing exhaustive search using all sorts of algorithms. There are things that are set up, just test using like really train multiple models, and there's ensemble models where you actually use different algorithms and you actually combine it together in addition to the ensemble models such as random forest. So I really don't have a good answer for that, but oftentimes like I do the lasso regularization because when I'm concerned about overfitting, I would try to go a little more minimal. Whereas if you have nonlinear, you expect nonlinear relations between your variable covariates and outcome, then you would go through nonlinear approaches, right? There's SVM with kernels, there's random forest because of multiple trees, then you have that, you can capture nonlinear effects. So yeah, I really don't have a good answer for that. In fact, one thing that I didn't toss in is deep learning. In my talk at Meetup, Data Science, I commented on one paper where they utilized fancy deep learning algorithm, and I did a journal club on it, but then I realized they already chopped up the sequence, and there's no use to you. It wouldn't add any benefits, right? So there could be overkill, right? And I used the same thing. I used the same data. I actually downloaded their data and run with deep learning. You need GPU to be faster, whereas I could just run on my desktop with lasso regular regression or ridge regression. The performance better than that one. But that's a specific case. It's not always. So there could be overkill. There could be, it's just a lot to consider. Yeah, I don't have a one answer for you. Yes?
Sorry, I'm not in biology. Of course, I'm in biological sciences, but I'm not too much familiar with genomics. So,
Yes.
what software do you use in your data analysis?
Right.
Like, do you use something like MATLAB, or do you
Right.
use, or is there a software actually called Genomics? Sorry for my background.
Yes.
I'm not too familiar with.
So that's a good question. Sorry, I forgot to put R on it. I extensively script in R, but when it comes to big data sets, it can get stuck, right? So sometimes I would use some essentially Linux command also to set an awk to try to deal with certain things. But in fact, there's also another really popular language which also has a lot of statistical and machine learning component nowadays, which is Python. So if you're just starting now, I would actually recommend doing Python. I still love R, but there's versatility. But really, R can do a lot of things already, and it can tap into deep learning packages as well. So, unless you're loading in gigantic data sets, which my co-op student is doing now, so it takes a long time, right? So yeah, so that would be my answer. I do R, but a lot of people do Python too.
Thank you.
Yes.
Hi, great presentation. I got here a few minutes late, so I'm not sure if you answered this at the beginning.
Right.
But I'm a grad student in statistics, and we're working with a lot of these ideas that you're talking about. But I have zero background in biology. So my question is, how much biological background knowledge do you need if you have the statistical and machine learning tools? How much biological background knowledge do you need to apply it to this field?
Right. So that's where I talked about jack of all trades and also collaborations. So it's a trade-off, right? So we can learn a lot of other things. Oftentimes, from my personal experience, I did have training in molecular biology, but I didn't have training in microbiology. And that's a matter of when there's collaboration, you have to keep up to date and learn whatever specific field, right? So this is a little more reversed learning where there's a need and then you move towards it. But it depends on whether you're trying to get a position in that needs biological knowledge, right? You can spend some time. I'll give you a tip that gives you a lot of comfort. I've heard from a professor in computer science telling me that it's a lot easier to teach a computer scientist who knows machine learning, the biology, than the other way around. So I really have to, and it really, because without analytical background, it starts from math, right? It's actually very challenging, and they have to learn to scripting and knowing the underlying math to know what tools to use, what analysis to do, what type of figures to generate. It really requires analytical knowledge. And so if you're more on the data analytical side, then just learn whatever things that you're interested in working on, and that would be my advice. But of course, we can get too much information, but there's always collaboration. So it depends on what path you're going, right? Even if you go into a company, I bet there would be experts in different fields. Like where I am, like I collaborate with microbiologists and I pick up a little bit of knowledge from time to time and also update it myself. Yes.
Thank you for the presentation today. So I want to ask about this microbiome similarity.
Yes.
I still hear that microbiomes are very like person specific.
Yeah.
So I wonder how similar it is between people from the same city.
People from the same city?
Yes.
Oh, for the individual?
Yes.
So what I did, the CAMDA challenge was the city, and the previous data was the disease. When I was doing the CAMDA challenge, there are a few papers talking about it. So they talked about how even though people in the same city, I think it was something about different offices, or like if you look it up, I don't know the details, but in general, different individuals' microbiome can differ. Like if we go back to that plot where, here it is. If you look at this plot, the first set is technical replicates. And so Bray-Curtis is an index for dissimilarity. The higher it is, the more different it is. And so technical replicates are more similar than biological replicates. And by biological replicates, in fact, it's the same individual collected 2 months apart. Okay, so that's why sometimes people say that microbiome can be identifiable to the person. And so it's just, there is variation, but it's higher than the technical replicates, but it's lower than intra-cohort. That would be, let's say, 2 healthy individuals. How different are they, right? So that's much higher here. And if you look at disease and healthy, it's a little higher than the healthy versus healthy, right? So just from this plot, you can get a sense of how different it can be from individuals. And I don't know if they collected these people in the same city. I have to ask my collaborators. But I'm sure there are differences. So again, rule of thumb, within an individual, with the timeline, it's more similar to a certain timeline. I think if it's longer, it can differ more. But between individuals, it can differ quite a bit. Because it involves diet too. Yeah. A person's diet can affect it as well. So there's just too many variables. Yes, Sylvia.
So maybe we can talk later, but just because you are on the same slide, I remember that you said something that, sorry to remind you that you tried something that you failed because of some of the samples were quite similar to each other. And I'd like to, if you can...
Oh, for the microbiome?
For this particular graph, yes.
Right, I should say it this way. So this is model performance, and it's called balance accuracy. But just think of it as the higher it is, the better the separation between the 2 groups, okay? And so what you'll see here is that MS versus RA, which is multiple sclerosis versus rheumatoid arthritis, the performance is much lower in this case. So when I try to do multi-class with random forest, I can predict multiple classes. It's like multinomial, not just regression, but with random forest, then that'll be predicting like 5 or 6 classes. And that didn't perform so well because things are too similar. And in this paper, my colleague actually, the conclusion is that there are some similarities in the gut microbiome pattern between those, among those with immune diseases. And because if they're similar, then it's hard to delineate them.
So again, I don't have much of a bioinformatics background, but when you were talking about the mystery challenge for
Yes.
this competition, so you said about the cities and the train cities were included in that program, but what if you get a data set that you don't have any train cities in? So how do you, what is the starting point of that?
Yeah, so if I don't have any cities, I don't have data, then I'm out of my job. I can't do my job. So it's like at lunch, I have an example. For example, if you just think of the DNA the crime group where, you know, if you want to find this criminal's DNA, you still have to match it to some database, right? So we always need that in order to do data science or analytics, because we need the baseline information in order to come up with an interpretation or a model. So when you have new information comes in, then it'll spit out its judgment, right? And so in this case, if it's like a criminal, you collected their DNA fingerprints and you want to match, it's not in the database, or if there's no database, you can't know who it is, right? If the database somehow has somebody relevant to this, related to this person, then you may track this relative down and try to find who that is. So again, it's a difficult question when you don't have the data sets there. And on top of that, this is difficult because we didn't have metadata. There's so many influence on microbiome. Like in the gut, there's diet, and in the environment, there's temperature. So one thing that I commented in the paper is that they picked the same day, which is summer solstice in the northern hemisphere and winter solstice in the southern hemisphere. And it's been shown that the different acidity in the soil, but that's soil. But in general, there are so many different environmental factors that can contribute to it. So, I mean, I don't have that information, and that makes it challenging too.
Great. All right.
Oh,
Okay, one
yes.
last question.
Hi, thank you so much for the talk. It was very informative. I just wanted to ask, so I noticed like in your references, okay, a bit of my background, I'm an undergraduate in computer science and I'm considering going to have a master's degree, but I don't know about a PhD. So I noticed that in your reference, everyone there has a PhD. And I'm just wondering like how important and how necessary it is to have a PhD from doing something like this.
Oh, I would like to connect you to our co-op student. So in computer science, it depends on whether you're going into academia, right? I'm not trying to pick only PhDs to work with. It's just, this is the environment I'm in, right? And in fact, with analytics, a lot of people take a master's and it can be really useful. They go into companies as well. And in fact, in our group, it's just the people that I work with tend to be domain experts of other things. But within our group, we don't really have that many PhDs either. Like they are really, really good at what they're doing. And so the key thing is with your computer science, you want to identify the domain you want to go in and then explore it while you're in grad school, right? As well as you can. And before you apply for a job, like before you graduate, continue to watch out for what are the job markets and what are the things that you need skill set that you may need to additionally acquire to reach to that job. So it's not necessary. Unless you want to apply for a professorship, then PhD is required. Otherwise, it's not.
Great question to end up with. Okay, so please join me in thanking Julie for a very well-rounded